Good morning, everybody. Let me first say a few words that this presentation actually is a result of. So this presentation is a result of the discussion between me and my mother about difficulty in the description of the processes in living substances. Using basically the tool, the modern theories that using modern quantum physics and other theories, if applied to the living systems, we can see that in many cases they do not describe, or it's hard to describe the observed phenomena in living matter. I will start with experiments by Eugene Makovski, who did these experiments in the late 70s. He studied the difference in properties of a living plant tissue and a dead plant tissue. So what he did, he exposed the living plant tissue to 200 atmospheric pressure. That's okay. He exposed a living tissue to 200 atmospheric pressure, and what he observed that the living plant just released some of the water, and it's remained alive, and it's retained, despite of this high pressure, it retained the water in it. When he performed this experiment on the dead plant tissue, under this pressure, all water were released, as if there is a difference between water in living plant and the dead plant, which chemically it's the same water. Once it's released, chemical analysis showed it's the same water. So what are the possible explanations of this experiment? So Makovski suggested, his explanation was that there exists some biostructure, in his opinion, that structured basically the living plant has a biostructure that structures water and help to sustain this high pressure. And he believed that when biostructure destroyed, when plant dies, biostructure destroys. There were one of the possible explanation when I was thinking about possible explanation, encountered the experiments by Pollock and his team, the experiments on easy water. They found that a zone they called easy, it's exclusion. Easy is abbreviation for exclusion zone. They found that exclusion zone forms on the border of the hydraulic material and water, and this exclusion zone has a different property than the rest of the bulk water. So, some of the properties of the exclusion zone, it has viscosity 10 times that of the normal water. It's also called exclusion zone because it does not mix with any other solutions introduced in water. It's about tenths of microns thick, and it also has a property that has negative potential, 150 millivolts with respect to the rest of the water. So the existence of this structured water can explain the Makovski experiment, yes and no, because easy water should form on both the boundary of the living plant and the dead plant. There shouldn't be any difference, but obviously we observe that living plant has a different properties than the dead one. Another possible explanation can be done if from the theory of coherent domains. That was a theoretical result. It was obtained by Pripata and Del Gucci in 1995 on the basis of quantum electrodynamics. They proved theoretically that water can, that coherent domains can form in water at normal temperature. We're not talking about low temperatures. At normal room temperature, they calculated that the volume of such domains can reach up to 40% of the total volume of water. It's like as if water exists in two phases. One is coherent phase, so all molecules in such domain are described by the same wave function, so you basically are dealing with the quantum objects, so it's become like two-phase liquid. And the stability of such domain, as they calculated, is due to the fact that the quantum energy level within the domain is lower compared to the energy level of the rest non-coherent water. So if such domains form in water, that can explain the Makovski experiment, and we can say that in the living plant, the water structures by the existence of these coherent domains in the living plant. But if we make this conclusion that coherent domains structure water, then we have to go further and say that living matter somehow can affect water at quantum level, because coherent domains, they are quantum objects. So if these coherent domains are formed in living plant, then we have to say that living plant affect water in some way that plant does not.
May I ask a question about 150 millivolts? Is that minus 150 millivolts with respect to the energy levels of water?
Yes, that's what I found. Yeah. Mm-hmm. So, but again, that's a very important point, that if we again assume that they are formed in living matter, only those coherent domains, then the question is by what mechanism a living plant can form or basically create favorable conditions for this domain to form. So there's definitely some difference between living and that. And if we think about living nature, the way it works, we see very many differences. Like we observe some molecular chaos in animate world, and for example, we talk about chemical reactions in non-living systems. They occur like via random collisions. But if you compare to the living systems, I show it schematically here, the chemical reactions in living organisms occur with the help of an enzyme. And if we follow the enzyme process, it looks like a very well-tuned mechanism. It's really very, it's the same process repeating itself over and over again. For example, just briefly how enzyme works. Enzyme is a chain protein. It's a protein, and it's fold itself. It's more like this. So it folds itself in a particular shape, and for a chemical reaction to occur in a living system, it should form the shape that exactly match the substrate. And the substrate just bind to enzyme in the place an enzyme called active site, and then enzyme it's like a catalysis of the reaction, and then the reaction occurs, chemical reaction occurs, and we should know that any mistaken binding of this protein in enzyme make enzyme inactive. And if we think, how can we explain this very deterministic, very precise process? And if you think about molecule by molecule, if you try to apply physics law, it's a quantum system, right? It's a quantum system, so it should be governed by probabilistic laws. But on the other hand, if we just try to estimate, if one tries to estimate the probability, for example, of a 10-link enzyme to fold in a particular shape, even if each reaction occurs with high probability, the error must accumulate with time, and if you calculate the probability of one enzyme to fold in a particular shape, it's quite low. But we know this reaction occur over and over again, generation after generation. So what we observe in living systems is quite deterministic, and it's hard to explain with probabilistic approach. Biologists very often say that this precision in the behavior of the enzyme and all these processes are due to in DNA. But to say that it's all hidden in DNA is like to answer the question with a question, because the stability of DNA itself is a big question. For example, so we know that conditions changes, the nutrient environment changes, but we know that reproduction continues over and over again with a high efficiency. And some facts, recent facts, there were experiments recently performed on bacteria Escherichia coli, and Abbott and Johnson Lewis, they showed that a single gene that encodes an average sized protein is estimated to suffer a mutation once at about million bacterial cell generation. That makes it very enormously stable. So if we will say that there are certain probabilities that the DNA is stable, then obviously we will not get such high stability. Also, another interesting fact about the stability of DNA that were obtained by Ackerman and Chow in 2006. They are screening the genome sequence of different organisms, and the screening revealed that the tendency for stability rather than for generation of variations influence how information is encoded to gene. So mutations are really unnatural. Gene trait the way gene is encoded it's more like towards stability than looking for variation. It's more like reproduction of the same thing rather than variation. So the stability of the gene itself is a big, interesting problem. So here I want to say that, as I said, this work is a result of the discussion between me and my mother. And on Monday she presented her work. And basically, first of all, I'll tell you that in our opinion, the problem with this description and this mystery can be resolved if the processes that occur in physical vacuum are taken into consideration, that the processes are not just the process we observe in the molecular level, but also in the finer level. It's some processes that occur in the physical vacuum that if taken into consideration, will resolve these mysteries. So on Monday she did a presentation in which she developed causal interpretation to quantum mechanics. And causal interpretation, it's always assume the existence of hidden variables. So in our view, these hidden variables are some processes in physical vacuum. And it follows from her equation that if those hidden variables introduced as the potential in the equations, then it's a mathematical result, and then as the electron moves in the atom, some kind of structures which must form in physical vacuum. So, and if that's the case, if atom, so let's say, have continuation in physical vacuum, generate some processes in physical vacuum, and molecule also generate some process in physical vacuum, and obviously biomolecule must have a more complex process, then the interaction between the structures that are formed in physical vacuum must be taken into consideration in addition to all these observed processes that in material world. That is just our explanation of that. So that this will solve this mystery if this is taken, if the physical vacuum is studied and the process are taken into account. Let me see, just one second. Okay, so just give me a sec. Yeah, I also want to talking about interesting properties that are observed in living matter. There are also there are several experiments done by different groups on transmutation of chemical elements in biological systems. It started in 1960s with the work of Louis Kerwan, who claimed that if you place living substance in some environment with a deficiency of certain element, then this living substance can create the, basically generate the transmutation of one element to another. He studied, for example, the processes of growing seeds. He analyzed 180 seeds, and he claimed that if you place those seeds in the calcium deficient environment, then he observed that as they grow, the level of potassium decreases and level of calcium increases, as if the plant within itself can change from can produce transmutation from potassium to calcium. So he studied this reaction, potassium 39 plus a proton with calcium 40. This kind of reaction are not observed in inanimate world, in non-living systems, and it's also occurred at room temperature. So also he did this experiment on different biological systems, on plants and also animals. He's one of the experiments when he did on trench fish, that he places fish in water that contain 1.4% of sodium chloride, and he measured the blood the potassium and sodium level in the blood of the fish, and he found that in the surrounding water and the level of the potassium and sodium in the blood of the fish changes as if fish generated this process of transmutation. Carvan believed that, he claimed that biological rules may not be directly converted in the rules of physics, because the rules of physics, according to him, were obtained based on the experiment on inanimate objects. But that was his interpretation. He made about 100 different experiments, but unfortunately his experiments were not very well explained, so I cannot really rely on them. But a group of Russian scientists, actually my mother worked with those scientists, and this group are very reliable. They were very precise in doing these experiments, and they described them very well. So they repeated some of these experiments, and they also observed the transmutation in microbiological cultures. For example, they tried to synthesize a very rare isotope of ferrum, ferrum 57, and they used Miss Bauer spectroscopy to detect this isotope, and they created a favorable condition for this reaction to occur. So, for example, they took a biological culture, they placed a biological culture in the solution, salt sugar solution that were prepared based on the heavy water, and they was studying the reaction when manganese salt plus heavy water deuterium synthesize ferrum 57. Ferrum 57 is isotope that is very rare in nature. It's about 2% out of all iron. And
Is it completely stable? Or
yes, it's a stable.
It doesn't have a decay channel?
No, it does not. 50, I think 58 is unstable, 57 is stable. 57 is a stable one. And what they did, so obviously they check, again, they did this experiment very accurately. They checked that that the glass material of the containers and anything that they used did not have this isotope. Again, they use Miss Bauer's spectroscopy that determines the presence of this isotope. They had, beside this favorable environment, obviously they had control containers. One was sugar salt medium prepared on the base of light water, regular water, and then just sugar salt nutrient medium based on light water with manganese salt. Again, they place biological, all this, biological substance in it. And the third was just sugar salt medium based on heavy water without manganese salt. What they observed that this biological substance that they place in all 4 containers, they observed that in the 4th container with the favorable conditions, they indeed, as a result of this experiment, found the presence of this isotope, ferrum-57, and no isotope were found in the other 3 containers. So that's proof that the reaction indeed occurred. So this is just from their book, from their paper. This is the result of the Miss Bauer spectroscopy.
That experiment is incredibly interesting, if it's true. Did they manage to get that stuff published?
Yes, they have a book published with several of these experiments, and again, my mother worked with this team. They know this team very well. They are very the trustable group of.
Okay, so
and they did a very good description of the experiments.
Unfortunately, I don't read Russian.
No, this book is in English. It's like they did it a very good. They made it in 2 languages.
There's a translation in English.
It's actually half book is in Russian and the other half is translated.
Yes.
Yeah. They have several experiments. This is was one that was easy to explain and present. The other may need more explanation. But they have several of these experiments. They also refer to the Karvaran and criticize his approach a little bit, explain what he did wrong. But they obviously provide their own explanation to that. But they're mostly a group of experimentalists.
Was that tried anywhere else? Has it been reproduced further? Especially this one is a very clear signature of IN57.
Do you know if it was repeated by any other group?
I think it was about 10 years ago, more than 10 years. But now I think somebody.
Okay.
Yes, I think.
Very interesting.
Yeah, I can give you the reference to this book.
Research was about 15 years ago.
Again, it's as I said, they create a certain favorable condition. They test. It's not like they place materials and then see what will happen. They test a particular reaction. So they create
in Japan, there is a group who is interested in this research. I know in Japan.
Okay.
So as I said, they test, the way they work, they test a particular reaction. They do not just see what happened. They test a particular reaction, they create favorable condition. Like for example, in the experiment that you just asked about, obviously you need a heavy water because you need deuterium.
So
you cannot do it with light water.
They're detecting Fe57 with mass spectrometry.
Miss Bauer.
So no Fe57 in the whole system before, Fe57 present afterwards. Is that what they see?
Yes. Obviously, the glass was. They in their description they said they checked the glass.
Yeah.
They checked everything possible that was used to prepare the experiment. Every single part of it was checked. Of course, they want to make sure that they have good experiment.
That's incredibly exciting. Why has that not been picked up?
It was, but as I said, I was in Japan, but we don't know. I don't know. Honestly, I don't know.
Could I ask the group as well, because I've forgotten my nuclear physics. I have a terrible memory. It's 50 decay channels. If I remember rightly from my nuclear physics about 40 years ago, the decay channels, the reverse process of that Fe57 potentially goes into something, pretty much are either gammas, neutrons, alpha particles, positrons, and that's pretty much it, isn't it? They never emit deuterons or neutron nuclei or protons.
You're talking about radioactive decay.
Yes, I am asking about it. Is the reverse process?
No. This is.
Oh no, I understand what that is.
No, deuterium here. There is no radioactive.
No, I understand. The question is, in nuclear decay, different subject.
There is. You're right.
Yes,
I think what you're saying is correct.
Yes.
Yeah, but this is more like fusion to produce
this is called nuclear fusion.
That's
This
right.
is called
Yeah,
nuclear
right.
biological fusion, if true.
Right.
Yes.
And again,
It's a fairly big thing.
what's amazing about this is that it's occur in room temperature.
In room
It
temperature.
does not
In the
require,
living
and
living
only in
organism.
biological
and it does,
systems.
right.
Yes.
It will never occur in the no, and it does not require any additional energy.
How do you know?
No, I mean there was no like
There's no
heating.
external source, but you
So
don't
there was
know.
no external. Yeah, I mean
Yeah.
yeah, that's what I mean.
Well, you can
There's
find
no
out.
external
It's quite
source.
simple. We know the mass of NN55 and of D pretty
Right.
closely, and of Fe57. Is there any mass? There will be a mass difference. You can find out whether it's
Right,
exo or endothermic.
they checked that, but I mean there's
Yeah.
no if you take
No,
it.
I know
in-
you're not putting an external source in, but one should look at
No,
the
what I
masses.
what I wanted to say that if
Yeah.
you even calculate it, you will need much more energy to produce it in non. If you try to synthesize
Yeah,
it
that's what I
in
suspect.
laboratory conditions, the
Yes.
energy you will need
I mean,
is
even if
much
it's-
more.
Even if it's 10 to the minus 18 grams, you're talking about huge energies for nuclear.
Right. That's what I
Yeah,
said. That
yeah.
you need for non-living. If you want to synthesize it, you really need enormous amount of energy to generate reaction like that. Of course, there was some experiments trying to see if that can help with removing radioactive materials from environment. They was trying to find some application to that. Okay, so. Let me see. So basically, as I said before, that if we go over some experiments, these experiments are just a few of the experiments that are done recently in biology, in the study of living substances in microscale. And in our opinion, with my mother, there is no really physics theory that explains it very well, all these processes in living substance. In non-living, we can explain many things, but in living substance, they obviously behave differently, and this difference is not picked up by, it cannot be explained by any known theory, any tools that we have. And as I said before, that in our opinion, the physical vacuum should be taken into consideration to explain these properties, and the information and some additional parameters that take place in these reactions are some process in the physical vacuum.
Sorry, as a layman who is not a physicist at all, can you explain what you mean by physical vacuum? How far does it extend? Where do we find it?
Again, it's only our view on that. That in our view, it is some sort of, probably some medium or some finer matter that have some structures in it. That's in our view. That there are. It's not. I'm not talking about contemporary physics, but in our view, it has some processes in it and some source of.
But the answer to your question is no.
No what?
No, one can't explain that. If you look at the consensus of physical opinion on your question at the present moment, no one cannot explain that. It's 123 orders of magnitude out if one tries, only, which is far larger than the number of particles.
Well, what's going through my mind is experiments or observations done by a Scottish guy whose name I cannot remember about the budding phases of plants and trees that are in sync with planetary cycles.
Lawrence Edwards.
Thank you.
Good man.
Is there an infection here? I don't know.
That's why we're here to discuss all this.
But the answer in terms of
Yeah,
contemporary physics is no.
Right.
Yeah, go ahead. Yeah, I'm not going to.
On the work of Pollock and Song, did you actually meet him in that group, or you
I
just...
met him, yes.
You did. Did you go to the annual meeting, the thing called WaterCon?
No, I just was present in his presentation
Okay.
at one of the conferences when he showed his experiments on
Well,
this
His
hydraulic
work,
gels.
his work on all the ones like Del Giusi before he died, they're online in great detail, and their fundamental idea is that anything which is alive has a cell, and the cell's got water in it, and that's why they're exploring easy water and stuff like that. And Pollock's written a book, it's called The Fourth Phase of Water.
Right, I know this, but then how would you, I'll go back with the question to you, but then what is the difference? Remember I started with experiment by Makovski, which clearly showed that living and dead plants behave differently under pressure. Easy water, according to Paul, should be exactly the same on the surface of on the membranes of the dead plant cell and the membranes of the living cell. So why do we observe the difference in living? Living is like, it seems more
Well,
structured.
if you restrict the answer to stuff understood by physics, there's no answer. But if you introduce some concepts out of living systems, I mean, they're exploring things like homeopathy. Luc Montagnier is there dealing with weird stuff which he got the Nobel Prize for. They're in another world. It's probably worthwhile picking up their recent literature.
I did read that book.
No, I didn't mean that. I meant the detail.
But it
one.
didn't provide me the answer
No.
why it's different in living and
Yeah,
dead.
if
Okay.
you go to Preparata and you see it's close to an answer.
There is a
But
simple
it's
answer.
in the area of quantum.
What Dilguch and Preparata are saying about easy water, they said that they believe that easy water is it's like a huge coherent domain. That's what they believe that form on the surface. But again, I'm coming back to the idea, if that's the case, why the living plant have a favorable condition for this kind of domain generated in it, and the dead obviously did not have this. So what's the difference?
Two reasons. First reason is a living thing is, if you look at the phase of water on a surface and start reducing the size of the surface, for example, we take a small tube and reduce the size of that small tube, where there's an interaction between the surface of the material of the tube and the water. Then for systems where the water is attractive, in other words something like glass, what one observes is it gets smaller, the point at which water melts, the phase change of water from liquid to solid increases. So there are tubes in your brain which are sufficiently small that the water inside them is frozen at the temperature of your brain.
But this mean that water is structured.
The water
At
is
a
structured
small-
within
at a
one's
small
brain, as
scale.
you suggest here, presumably in the easy phase transition. That's the first thing. The second thing is 150 millivolts is a lot. 150 millivolts is a very great deal in
Yeah,
chemical
right,
reaction
right.
terms. It's 10% of typical big molecular transitions. So when one's talking about a negative potential for living things of 150 millivolts, that would make a huge difference to the structure of that water and to the way that it's put together. Now, if you're looking at a crystalline structure of water, in other words, an ice, then that would have exactly the properties that you're talking about, and that ice could easily be formed by, because water is a polar molecule, by some extra ordering that's produced by an electric field. So the next experiment to do is to try taking a membrane, putting a voltage over it, and seeing whether or not that then has the properties just with a voltage. that
They
you observe in
believe, yeah, they
living
believe that
things.
voltage actually
So do I think
help
that's-
with this
yeah, I think that's
electronic
really necessary.
oxidation.
But also we know the structures in the brain now are very complex in the water nanotube domain. So, there's a doctor in Germany whose Sanskrit name I remember, but I can't remember his real name, sorry, his public name. He's called Manohar. Anybody know him in Sanskrit? I know, anyway, we can get in touch. He's the guy who taught me about this stuff. But there is also a literature on that in the medical areas coming up.
So
So
what then?
I don't know if that's Pollock or not, it may well be. Was it Pollock, Mike? The chap who was doing easy water?
Yeah,
He-
Paula.
Okay, good, yes. I don't know whether that contains that or is in touch with that literature or not, but that's where I got it from.
So what's in your opinion then, what happens when this plant dies?
You lose the potential. Stuff falls to pieces, the water's no longer bound to the surface.
But it doesn't fall in pieces. That's a problem, that it's remained the same
Oh,
in
yes.
terms of molecular structure.
No, the water structure falls to pieces, not the plant,
So why? That's a question,
because
why?
the potential is missing.
So what caused the potential then? No, they
Life.
found, They found the potential, Pollock found the potential difference in non-living. He was doing experiment on hydrophilic gels. He found this potential in non-living. It
Yes.
does not depend on if it's living or not. That's a good question.
Yes.
That's what I want to understand.
Yeah.
Why living and non-living is the same potential, everything's the same. What's the difference? What happens when the plant dies? That's what I want to go understand.
Okay, so
Add something here, yeah, sure. I can
Translate
translate.
me because of my English. Если мы добавим внешний потенциал, уравнение
А я
Шрёдингера,
сказала. So she's talking about Schrödinger equation. If it,
Это не отметает задачу, что только probabilistic approach, и нужно все равно искать новое поле. Вот я ему говорила, это не так нужно.
okay, that's okay. She came in too late, and she said that I said it already that
Okay.
if potential is introduced in Schrödinger equation, then we don't need to have this probabilistic approach, and that's
We
that.
need
The additional field to explain this processes.
Yes.
It's a different field in both cases,
Different.
or not? Okay,
Different.
that's what I wanted to establish.
Yeah.
That difference can make a big difference in the tunneling probability. For example, if you do a Schrödinger transition probability, a millivolt can change stuff at a volt
Mhm.
by a lot, right? Right. So
Yes,
that
yes,
can make a big difference.
yes,
Can I ask about fat? Because you've got to have fat for a cell.
Uh-huh.
So where does we're sort of talking about the inside, but what about the boundary? Where does the boundary, how does that relate to this in the vacuum?
The boundary.
Well, the fat, the layer of fat, the lipid bilayer around.
So again, it's only our idea. So in our view, like that's what my mother mentioned in her presentation, that we believe, for example, from her equations, it follows that if electron is moving in the atom, it must generate some kind of precession, some kind of structures and physical vacuum.
Yeah.
So in our view, basically in a very rough model, you can say that every quantum object has some continuation physical vacuum, generates some
absolutely.
process, and then these structures also interact with each other. And obviously biomolecule must have more complex structures.
So, I mean, what are you saying then that cells are absorbing aspects of the environment, aspects of the structure of the environment?
I see what your question is.
Yeah.
So in our view, that living substance in some way, yes, can affect what in quantum level through physical vacuum structures,
Yeah.
what, and there are some, that vacuum has quantum properties, and this interaction occur in vacuum.
There's
It's
a
a quantum
there's a
concept.
biologist who in UCLA who I've been talking to who's very interested in Peter's work and very
Mhm.
interested in the physical connections to biology. And he
You
is...
understand why, because there's no answers right
Yeah,
now.
no, absolutely. That's exactly his point. He said biology as it stands is really a science. It's just describing things all the time, but it doesn't have a mechanistic
That's
understanding. But his view is that if you, I mean, he's particularly focused on lipids, so he's saying, well, actually these molecules occurred under particular environmental conditions, and things like cholesterol must be, they're kind of fossils. They could only arise at particular moments in evolutionary history. And so the cell gradually sort of evolves into, you know, us.
Right.
But you've got to somehow we've got to factor in not just environment but history, but time as well in that process.
So again, it's just the hypothesis, but our hypothesis is actually, my mother's hypothesis is that, again, it's just hypothesis that this information about the processes might exist in physical vacuum even prior in some way
Yes.
to these processes, and
Yes.
they can be like basically everywhere.
Yes.
And then these processes are basically originated
Okay.
from
So what-
vacuum.
So you get a cell.
It's like in some, I don't want to call it information because it's such a term,
Yeah.
but basically information about the process
All right.
partially
But
is
the
in
ce-
vacuum.
Cells are talking to each other, aren't they?
Yeah,
So-
and how? The question
Yes.
is how. That's
Yes.
my question.
Absolutely.
And our answer is through vacuum.
Yes, and actually the problem seems to be when you look at the chains of proteins and enzymes and all the rest of it and the way people describe it, you think, how the hell does this hold together?
Right. And again, if you compare to in animate world where you see all this molecular chaos, you don't see it in living.
No.
It's like well-tuned mechanism, just work after generation after generation.
I think I might put you in touch with this guy.
Yeah, but that's exactly what you're saying, that how they talk to each other, basically. That's what we are thinking, that it's through vacuum and processing vacuum, and that's additional,
Yes.
should be added to physics as maybe additional field, like my mother's.
Is there an inclusion zone as well, like on the other side of the boundary, the internal?
No,
It's
exclusion
just on
zone,
the outside that you'd find this layer of the property of water that sort of, or is there some interesting thing happening on the inside of the boundary as well?
No, what Pollock found is this particular, they called exclusion zone because it doesn't mix with anything. That's
Oh,
the
I see,
problem.
I see, that's
And then
what-
they study the properties, like it was mentioned in his book, they study properties and homeopathy, everything, like just how does it, this layer, they believe this potential difference, actually, that's what causes oxidation reduction of these processes in biological systems. Yeah, they explain, but to answer your question, no. They only found this interesting layer on the surface.
I've just got a question.
Oh, yeah, sorry, I didn't.
Excuse my being daft. You're describing two things here. One is a coherent zone of water molecules, and the other one is some cold fusion reaction of manganese into iron.
Yeah, it's all different
They're different
things.
things.
Yeah, different things. Yeah, sorry if I...
This is electrically two weird things in it.
Yes.
So yeah, I'm just throwing into you some
Okay.
interesting
Right. I understand.
experiments that
I was
work
trying to
on
connect
with
to
biological systems, and that shows that they really behave in an interesting way which cannot be
And
explained so easily.
how much iron 57 was actually made as a proportion of the weight of the maximum ball?
Probably very little. I don't know exactly, but probably very little. They just found no traces of iron.
It's too many.
Yeah, I cannot.
Even one?
Even one would be
atom?
astonishing.
Yeah,
If
it's not like
there to
the
begin with,
amount.
then it appears.
It's-
Doesn't need a lot.
Yeah, that's
Yeah.
exactly true.
Yeah.
Basically, we just need to see where
See
the reaction
that it
occur or
exists.
not.
Yeah.
If this occurred, that means if it was not present and now it's present, that means
Where
it's
the hell is
mutation.
that? If that's true, that's astonishing.
Exactly, it's not like
Yeah.
Okay, next.
Okay, thank you very much, everybody. I'll stop here.